In this video, I would like to summarize the review article entitled Bone microRNA and Aging. Aging is characterized by a steady decline of tissue functions. This is a consequence of a number of biological processes, also known as the hallmarks of aging. The aging is initiated by the primary hallmarks, or more precisely, by age-related epigenetic changes, loss of proteostasis, and telomere attrition. The primary hallmarks are followed by antagonistic hallmarks. The antagonistic hallmarks are beneficial in physiological conditions but become deleterious in aging. They consist of cellular senescence, altered nutrient sensing, and mitochondrial dysfunction. The final outcome of the above changes are integrative hallmarks, which directly affect the tissue homeostasis. One such outcome is the exhaustion of stem cell reserve. All of the hallmarks of aging are regulated also by microRNA molecules. MicroRNAs are 22 to 25 base pairs long non-coding RNAs that specifically bind to messenger RNAs and mark them for degradation. They are transcribed from the genomic DNA and regulated by the same promoters as genes. The methylation of promoter regions in genomic DNA can directly affect the expression of protein-coding genes and microRNAs. When the promoter is not methylated, the microRNA, shown in yellow, can be transcribed and repress the expression of its target genes. If the promoter is methylated, the microRNA is not transcribed. More often, microRNA molecules repress the expression of methylation enzymes such as DNA methyltransferase, abbreviated as DNMT. The messenger RNA for DNMT, shown in red, is in normal conditions translated into the protein. If the specific microRNA, shown in yellow, binds to DNMT messenger RNA, the transcript gets degraded and the protein levels of DNMT are reduced. This leads to general suppression of DNA methylation. Each cell also has a biological clock known as telomeres. Telomeres are the extreme regions of chromosomes and are slightly shortened during each cell division. When they become critically short, the cell stops dividing and becomes senescent. Stem cells express enzyme telomerase reverse transcriptase, abbreviated as TERT, which restores the length of telomeres and prevents the senescence of the cells. The expression of telomerase declines in differentiated cells. Some microRNAs can target telomerase, which leads to premature shortening of telomeres. If the affected cells are mesenchymal stem cells, then they stop dividing and cannot differentiate into osteoblasts. This depletes the osteoblast pool and leads to reduced bone deposition, which decreases the strength of the bone. MicroRNA molecules can also leave the cell and enter surrounding cells, where they can induce senescence. But the good news is that senescent cells can be rejuvenated by reactivation of telomerase. This was shown on cartilage tissue in osteoarthritis, where restoration of telomerase activity caused restoration of cartilage function. Mesenchymal stem cells with greater telomerase activity are also more likely to differentiate into osteoblasts and have higher osteogenic potential. This can be leveraged for therapeutic purposes. The rejuvenation of mesenchymal stem cells by reactivation of telomerase can restore the function of osteoblasts or chondrocytes. This can be achieved by exogenous expression of telomerase or by reducing the activity of microRNAs targeting telomerase by using microRNA antagonists. Indeed, studies showed the potential of microRNA-coated nanoparticles to restore the bone strength in mice. Some take-home messages. Aging is a set of physiological processes also known as the hallmarks of aging. Telomeres are biological timekeepers and determine when the cell stops dividing and becomes senescent. Reduced bone strength is related to higher numbers of senescent cells. Cells can be rejuvenated by restoration of telomeres. This shows great potential for therapeutic intervention. Thank you for watching this video presentation. Please read the whole article for in-depth information about the role of microRNAs in bone aging.